So I want to talk about something dear to my heart, and that is using DNA to look at biodiversity. But as a biological anthropologist, I sort of specialize on looking at primate DNA. And traditionally, one of the goals that we have always had is to name the various groups of primates. We have to know what we're talking about and give them labels. We usually would use their morphology, their skeletons, the color of their fur, the shapes of their bodies, and all of those things, maybe what they ate, who they mated with, and all of those things. But for the last 30 or so years, we've been able to use DNA to try to sort of elucidate their evolutionary history. Back in 1997, we first, a group of graduate students and colleagues and I, were able to get DNA from these particular chimpanzees found on the border of Nigeria and Cameroon. And it turned out their DNA was very distinct from that of all other known chimps. And as a result, we proposed a new subspecies of chimpanzee, Pan-Troglodytes elliottii. Now, this was very important because all of our laws, our Endangered Species Act, the CITES treaties, and other international treaties that deal with endangered animals, they need to name the groups of animals, and they need to know where they live and how diverse they are so that we can try to put our efforts into conserving them. And so this was one of the earlier uses of DNA to actually define, in this case, a new subspecies of chimpanzee. The problem is, even though we have these powerful genetic technologies, there's no direct measure of what is a species or what is a subspecies. I can't tell you two populations that are X% different are subspecies and Y% different are species. Much like the law, we have to deal with precedent. So what we typically do is we look at well-accepted arguments. In this case, species A and B are accepted by the entire scientific establishment as being separate entities. Now, we've measured two new populations, say population 1 and 2. Because they are more distinct than the already defined species A and B, we're very confident then that we can propose that populations 1 and 2 would actually be separate species. And this is typically and very commonly accepted. On the other hand, sometimes we find that the two populations, like 3 and 4, aren't very distinct. And so we have to say, well, they don't rise to the level of species, and maybe we can look more closely to see if they're at the subspecies level. And we can do this sort of transitively down and up the levels of sort of the evolutionary tree to try to get the proper names. And again, the names are really important because they are what are used in legal, in various laws and in making conservation decisions. An excellent example of this process was just carried out 2 summers ago with some researchers in my lab and elsewhere around the world who carried out some very grueling field work in the Congo Basin. And they found this cute yellow-faced monkey there that you see, which in many ways, except for its color and a few minor differences, looked somewhat similar to the guy with the bright white nose there. And so the question arose, is this indeed a separate species, or is it just sort of a yellow-faced version of the guy on the other side of the slide? Well, carrying out DNA analyses of segments of the Y chromosome and of the X chromosome, it was quickly realized that these two monkeys living in the same region were more distinct than numerous other species of monkeys. And so this is one of the new species of monkey, Cercopithecus lomamiensis, that was discovered and named in the summer of 2012. That's clearly an absolute distinct new species, not just a subspecies, but a new species of monkey that had been, heretofore unknown to science. And it was a combination of their field work there, but it was especially the DNA analyses that allowed us to draw these conclusions. In 2003, we had done similar work looking at gorillas found in the Cross River region of Nigeria, and it turned out they were incredibly distinct from all other gorillas that we knew in West Africa and Central and East Africa. And a new subspecies of gorilla was basically defined by genetic research, later verified by ecological and other behavioral research. And this is gorilla diele. So what was interesting is this gorilla, we got the DNA from these gorillas out of their feces. And in fact, that chimpanzee that I showed you, all of the DNA we had for our initial work was solely out of hairs that they shed and we found on the forest floor besides their nests. Now, being able to characterize animals by their hair and by their feces is really important. You don't have to trap them. You don't have to dart them. You can non-invasively get their DNA, and many of these animals are critically endangered. Unfortunately, many of them are hunted by people in the region, and so we don't want them to become familiar with us or familiar with people. We want them to flee from people so that they're not killed for meat or in pet trades and other things. So the ability to collect this DNA non-invasively has been probably 3/4 of the research that we've been carrying out in my lab over the last 22 years. We can do all sorts of things with this non-invasively collected DNA. For instance, these gorillas are very elusive. For the first decade, that gorilla diele, we had one fuzzy picture of it running into the forest. It took many, many years to get clear pictures of these things so that we really knew what they were like. And the way we would try to figure out how many are there, and there's unfortunately only hundreds of these particular subspecies, they would go around and count the nests. So gorillas, every night when they go to sleep, they make a nest. And so if you wander around the forest and you find a group of nests, you can count how many nests there are and get a rough estimate of how many gorillas are in that population. And then you wander around for several more days, you find another nesting site, and if you find 10 in one, 11 in another, 12, 11, 10, you can guess that you probably have around a dozen animals. But unfortunately, sometimes 2 of them will sleep in the same nest, or he'll wake up grumpy and make a new nest. And so just counting nests isn't good enough. But remember, you're not even seeing these animals. They're hiding from you, and we want them to hide. One nice thing in particular about gorillas is they eat a very strong fiber diet. And so one thing that almost probably 99% of the time when a gorilla wakes up in the morning and wanders off to start eating, they leave a DNA sample behind for us. And so by going out in the forest with our little pooper scoopers and scooping up samples from one nesting site and then a day later another site and another site, and then we DNA fingerprint those sites, not only can we count the number of unique individuals there, we can figure out their relationships. We can see that the male silverback has fathered all of the offspring. We can see if the females are related to each other or if there are other related. We can come up with an entire family tree and an accurate count of a population of a group of animals we have not laid our eyes upon, just by collecting the data that they leave, the samples that they leave behind. So there's all sorts of parameters of a population that we can gather by collecting genetic information, and these are very important to us in making our conservation decisions. Is this a unique population? Does it have enough genetic variation, or is it too inbred to survive? Where do we want to put our rare resources into trying to conserve things? And we can collect lots of DNA samples to answer many of these questions. We can figure out what they're eating. You can imagine a monkey 30 meters up in the tree, no matter how strong your binoculars are, you're not going to be able to see every insect and every fruit and every type of leaf that they eat. But gravity will bring the DNA of everything they ate back down to the forest floor when they defecate. And so by sequencing all of the DNA in their feces, we can actually accurately figure out all of the things that they've eaten in the last day or two. This is important again in studying biodiversity. We can even determine the proportion of the different foods they eat. If they're eating 60% of one thing and 20% of another and 10% of something else, from the DNA of the food left behind, we can actually figure out aspects of their diet, not just what's in it, but the direct proportions that are in it. And all of these things, collecting this non-invasive DNA, falls under the broad new category we call environmental DNA. Organisms leave tons and tons of DNA and biological samples behind. We respirate, we salivate, we urinate, we defecate, we menstruate, we ejaculate, we depilate, we exfoliate, all of these things. And ultimately, we decomposate. I couldn't figure out a way to get decomposition, but when your body goes back into the soil, the DNA in it is released into the environment as well. So if we can go out there and collect that DNA, we can understand what's in the environment. And that's, again, what we call environmental DNA. We can identify species and subspecies, or maybe more broader, the genera and orders that live in a region. And we have all sorts of techniques for trying to figure this out in the laboratory in conjunction with field work. So I can send somebody out on the savannah and dig up a few grams of soil from here and there and there. We can bring that back to the lab, and we can actually then determine all of the animals that have been running around the savannah for the last few days or weeks from the dirt, from the DNA left behind, from all those things that they've excreted that's left behind in the dirt, because we have these very, very sensitive techniques that allow us to determine what was there. Now, DNA technology, much like the cell phone and the camera technology that you just heard about, has leapt by leaps and bounds. It has even outdistanced the growth of computers and computer chips. So using what we refer to as next generation and second generation DNA technologies, we can go from the simple technologies we used in the late 80s and the early 90s to up to today, collecting hundreds of thousands of times more data in a given amount of time and for a given amount of money than we did just a few years ago. 2007 was a real inflection point in DNA technologies. For the longest time, DNA technology basically followed Moore's law of computers, and that is computers double in their capacity on average every 24 months. But in 2005 through 2007, some new technologies were invented so that we can go to, from sequencing 1 million bases of DNA, which would have in 2007 cost about $1,000, we can now sequence 1 million bases of DNA for a dime. And so we now collect billions and trillions of bases of DNA from very, very minute quantities. We've sequenced the entire genomes of Neanderthals and other extinct animals from the DNA left behind in their bones. So again, going out there to the forests or the ponds, rivers, and lakes, we're able to see everything that has been out there using these new technologies. One interesting study actually went out on the savannah and not only identified everything that was there today, by digging down just a few centimeters, they were able to see what was there last season and 5 seasons ago and even 10 seasons ago, and in fact the relative proportions of the animals that were living there. And again, going back in time, figuring out the biodiversity of a region would be very useful information. There's another source of DNA that's also lumped into environmental DNA, and that's biting insects. I now love mosquitoes because I think of mosquitoes as 1 million little hypodermic needles flying around the forest, sampling every animal out there with blood. I can go collect 1,000 mosquitoes, mash them all up in a blender, get the DNA out of all of them combined, throw them into these new technologies, and discover the dozens or hundreds of animals that they may have been out there and bit. I might have had to send a graduate student out for 6 months to see a particular species of monkeys in a forest, or a nocturnal lemur that's very shy. Mosquitoes likely going to find them eventually. And so by sampling enough mosquitoes, we're going to get blood from basically almost anything that's out there in the forest. And so this is another exciting technology. Last summer, we actually used, again, harking back to the previous talk, a drone flying over a swamp with a net to capture mosquitoes to sample the DNA of the animals in that region. So there's all sorts of neat things we can do. You can go, instead of doing it out of the soil, you can do it out of a pond, a river, a stream, or even the ocean. And so you could even begin to look for animals that you don't know are there. So imagine, for instance, a hypothetical. Maybe in Loch Ness, there is an unknown animal. Well, if you sequenced a couple dozen parts of Loch Ness and grabbed a liter of water and filtered it down, got all of the environmental DNA out of that particular sample, I'll bet you a six-pack that you will find an unknown species. It's probably not a plesiosaur. I'm guessing it's going to be a fish that you didn't know lived in that region, or a new species of snail or mollusk or something like that. But in a body of water that big, I'm sure there are unknown species. And so last summer, I headed out to the Pacific Northwest with sort of the same goal in mind, that I would use environmental DNA to help people look for an unknown species. In this case, Spike Television brought me in as a consultant, a scientist, and the co-host of a show called $10 Million Bigfoot Bounty, in which they had teams of people scouring the Pacific Northwest for evidence of Bigfoot. I designed and built a portable DNA lab that in 6 hours I could sample 100 different biological samples and determine what species were out there. So they'd say, this is scat from Bigfoot. And I was like, well, that's actually a coyote. Oh, we found saliva on feathers from Bigfoot. No, that was a mountain lion eating a blue jay. But nevertheless, we were able to apply the very same techniques I use in my lab just across the park out there in this portable lab in the field. So you might ask, why did I do this? Well, the themes of this conference: speak, connect, and seek. We sought something. I then went on television and do something I think is very important, and that is push science to the public. I got to discuss the scientific method, how evolution works, how DNA technology allows us to identify things, the new technologies that we're using. I got to talk all about sort of science outreach to hundreds of thousands and even millions of people through the rather interesting venue of the search for Bigfoot. Well, I don't want to give a spoiler in case you haven't seen all of the show. Let's just say that the other reason I did it, it was fun. And if what you do, if science isn't fun to you, don't become a scientist. If filmmaking's not fun, I wouldn't become a director. You need to find something out there that you're passionate about and that's fun. Thank you.